Hello, I'm Nicholas Newell, an independent researcher in structural bioinformatics. My poster concerns beta turns, which are the most common type of local structure in proteins after the alpha helix and beta sheet and play key structural and functional roles. Turn backbone geometry has been classified at multiple levels of precision, but the current picture of side chain structure and interaction in turns is incomplete because the distribution of side chain conformations associated with each amino acid sequence motif in turns has commonly been represented only by static images of typical structures, and only motifs which specify single amino acids have been systematically investigated. Furthermore, no general evaluation has been made of the side chain interactions between turns and the structures in their backbone neighborhoods. Finally, the visualization and comparison of the wide range of turn conformations has been hampered by the almost exclusive characterization of turn structure in the Ramachandran space of backbone dihedral angles. This work introduces MapTurns, a web server for motif maps, which are interactive 3D graphical conformational heat maps that employ a turn-local Euclidean space coordinate system to comprehensively map the distributions of backbone and sidechain structure and H-bonding associated with individual sequence motifs in turns in the backbone context. Motif maps are generated from a redundancy screen PDB-derived dataset of 102,000 turns by a three-stage hierarchical clustering procedure. Maps characterize many new sidechain motifs, provide rationalizations of sequence preferences, and support mutational analysis in the general study of sidechain interactions, and they should prove useful in applications such as protein design, which can benefit from the comprehensive and detailed picture of structure and interaction which they provide. MapTurns is available at www.betaturn.com, along with a broad map-based survey of sidechain motifs in turns. To demonstrate map turns, we can use the motif finding tools to identify the most important motifs in the largest beta turn type. We can select the turn type, and we see the backbone clusters that correspond to that type. We then bring up a list of the important motifs within the type, and we can select from the first order or single A motifs. We can choose aspartic acid at the first turn position and copy it into the map display box and display the map. The map shows backbone clusters on the left and sidechain clusters within them on the right. We can select backbone clusters using the menu entries. And as we do, we can see it shows us the distributions of sidechain clusters within each backbone cluster. The menu entries are ordered by importance so that we can quickly access the most important conformations. We can choose from the sidechain clusters using the sidechain menu. We can see that the most important sidechain cluster has an open loop context, but the third most important cluster has a context which shows more structure. If we open the second level map page, we can explore the structure of this context. So we'll select the third sidechain cluster. And now we can choose from the tail clusters, which represent the context of this structure. Next, we can browse the structures that we have selected. And we see that structures correspond to the type 1 beta bulge loop. It is a very common structure. To identify the most important sequence motifs that occur in a particular backbone context, such as a hydrogen-bonded loop, we can use the motif finding features of Explore Turns, a tool also available at betaturn.com. For example, if we want to find and map the important pair motifs that occur in the type 2 or 6C1 beta bulge loop, we load the motif in the tool and enter the wildcard motif string that ranks all pair motifs at the positions the loop occupies. We then copy the most significant motif, which is a cysteine pair, into the sequence motif box and open its map. The map for the cysteine pair, in which cysteine occurs just before the turn and at the third turn position, shows that the motif occurs most commonly in a flat type 1 turn geometry. The sidechain cluster distribution within that backbone cluster shows that in the two largest sidechain clusters, the cysteines adopt a specific orientation. To investigate the context in which this occurs, we open the lower level map page. When we select the largest sidechain and C-tail clusters, we see that the hydrogen bond heat mapping shows that the pair of H bonds characteristic of the 6C1 beta belt loop occurs with very high frequency. And when we browse the structures, we see that the cysteines are coordinating metal ions. This structure is the rubidoxin knuckle found in zinc fingers, which function as interaction modules that commonly bind nucleic acids and other molecules.